So in this video about cells, I just wanted to cover with you what I expect you to know for the quiz and the exam. This is the cell. If we say cell, remember, oops, try again. This is a cell here. Sorry. And this is the cytoplasm. This is the internal environment of the cell. And this is the extracellular fluid. So this is the external environment out here, and this is the internal environment. So since cells are metabolically active, they need to have energy, and they need proteins, carbohydrates, sugars, vitamins, minerals, water. Now remember that every cell also has another job to do. It can be a cell that creates hormones. It can be a cell that contracts like a muscle. It can be a cell that moves along an action potential like a nerve. These cells have many different functions. So we want to talk first about how these cells get fed. And I want to explain that most of the cells in your body receive their nutrition from a nearby capillary. And there's a very small distance between the capillary and the cell. Now capillaries are one cell thick. So this tube, which is the capillary, has only a single cell, one cell thick. And it has small openings, and this can flap. So the pressure that comes, say it's coming in this direction, the pressure, it will push plasma out into the extracellular fluid. This is called filtration. So that's that dinner that you ate and you digested, and it went through the liver and got processed, is now in the general circulation. So the proteins and carbohydrates and sugars and vitamins, minerals, and water are now being pushed out into this extracellular fluid, so now it will be available to the cell. So understand, the cell doesn't have a mouth like you have, so it has to have a different type of mechanism in which to eat and drink. So the first thing I want to talk about is the membrane, and this membrane is called a plasma. Look over here. This is a plasma membrane. Okay? It has these proteins that float around the top, and these are receptors for the most part, and they're also flags with an identification card. It's called an MHC marker. You don't need to know that right now, but you'll learn that in physiology. This tells everything else in your body that I am a self cell. For me, it says I'm a Jackie cell. Okay, that's so that your white blood cells can identify it and it won't destroy it. Okay, I hope that makes sense. Now if you'll notice these little molecules right here, this is what the membrane is made of. It's a circular head like this with two tails. This is called a phospholipid. That's all you need to know. This part is polar, So it loves water, so it can. So you want the water right up next to the membrane, so that it can bring these molecules by diffusion, right? Diffusion. Oops, D. We'll go down a concentration gradient and into the cell. I'll talk about that in a minute. Okay, phospholipid. These have a tendency to arrange themselves all by themselves in a circle called a mycel. You don't need to know that. If I build these in a laboratory and I throw a bunch of them into a bucket of water, they'll make a circle with all their little tails on the inside. So it's a naturally occurring conformation. So the cell membrane is kind of easy to understand in that way. Now the other thing is, which is kind of interesting and important, is that there are molecules embedded, and they kind of go like this sometimes. I've seen this depicted in different ways. This molecule is cholesterol. Might be surprised to find that out. This gives structure and support to the membrane so it doesn't tear apart. Now, so that's what I want you to know. So with all of these proteins here, the entire thing is called a fluid mosaic model. I want you to know that. The model, fluid mosaic, of the cell membrane, the plasma membrane, is called the fluid mosaic model. If you look at it, and the videos below will show you, this is constantly undulating because it's floating in water. It's just floating. This membrane is floating. And these membranes, these float on top of these molecules. They float around. They move around. Constantly moving, constantly moving. These are receptors. They're looking for drugs, hormones, things like that. We can say that this is insulin, right? Or an insulin receptor. So if insulin comes through here, you've eaten a big sugary meal, and an insulin molecule comes and it looks like that, it will fit right in here and it will trigger this integral protein to change its shape and move sugar into the cell. And that's how the cell controls how much sugar comes into it. This is a very, very specific receptor, a specific protein. It's an insulin receptor. It's a good example because everyone understands that. I know I got a little bit ahead of myself as usual. So the way that things get into the cell are the following. First, there's passive transport. And that's diffusion. That's small particles and like oxygen and ions down a concentration gradient. Osmosis, this is water. And then it has facilitated diffusion. Which is using, this is called an integral protein because it passes through both layers of the membrane. So this is an integral protein, and this allows for facilitated diffusion. Okay? Facilitated diffusion. Down a concentration gradient. Things can also move out down their concentration gradient. And sometimes these protein channels are specific for a specific molecule. Okay? So that's passive transport. And then, I don't know if I have an eraser. Let me just hide. So, that was my cat. Go back here. So that was passive. So active transport is moving things into the cell against a concentration gradient. And concentration can be abbreviated that way. Gradient. Okay? So now we have, first of all, the molecule that needs to go inside, needs to be eaten by the cell, has to be recognized first. So we have these are receptors, right? And they can recognize when a molecule that they need can be passed into the cell. So we always need some type of integral protein, right, which is usually closed, but it has a specific receptor and it has a shape and a charge so that only the molecule it wants to get through can get through. So this is against a concentration gradient. And these are things like pumps, like sodium-potassium pumps, extremely important in nerve cells. Pumps, endocrine hormone receptors, to allow sugar in. Okay, and then we have exocytosis. When a cell manufactures a product, it needs to get that product into the extracellular fluid to be picked up by a capillary and taken off to the rest of the body. Or it can eat by exocytosis. Exocytosis is when you want something to go out. So let's say the cell manufactured a packet of hormones. Say it's insulin even. That will move towards the membrane, and when it reaches the membrane will go like this, and it'll pinch off that vesicle, and then this will go, and the vesicle can break, and then the molecules will be picked up by, there's a capillary over here as well, and those molecules will be picked up by the bloodstream and taken to the rest of the body. Now in endocytosis, this is how the cell eats and drinks. Endocytosis. Eating is called phagocytosis. And drinking is called penocytosis. And it goes like this. There's a molecule here. It comes up to the membrane. It fuses with the membrane, and then the membrane pouches inward, takes this vesicle, and then it pinches it off and it moves inside. Then you have activities that happen, chemical activities that happen within the cell that will break this down. And if this is protein, it can eat it. If this is a white blood cell and this happens to be a bacteria, it will digest it and neutralize it. So basically, this is how things move in and out of the cell. This is filtration, how the capillary bed feeds the cell. The cell can only eat through these channels. Or, in the case of passive diffusion, they can pass right through. These are very small molecules like oxygen, carbon dioxide, water. Sometimes a little bit of water can pass through, or small uncharged molecules. Okay, so this is why the membrane is so important. It protects the cytoplasm and the internal environment from the external environment. If it were open, then all of these processes couldn't happen. So it is the living component of all organisms, and it's teensy, teensy, weensy. So this is capillary function, filtration, the pressure behind the flow of blood pushes plasma out into the extracellular area. So the fluid inside a capillary is called plasma. When it leaves the capillary, now it's called extracellular fluid. And the closest layer of fluid right here is actually called, interstitial fluid. So if you see that word anywhere, interstitial fluid, it's the closest fluid. Fluids are all over your body, and depending on where that fluid is, it's called something different. That's how anatomists and physiologists can identify which fluid you're talking about. So in the I think the next video, it goes through the organelles, and it goes through them very, very well. Okay? Like this is the nucleus. So just learn the organelles, the names of the organelles and what they do, very simplistically. I care more that you understand how things move in and out of the cell. You'll see videos that will name these particular proteins like carrier proteins and receptor-mediated proteins. I don't care about any of that. They're all integral proteins, and they limit the passage of things in and out. And they're very specific about what they let in and what they let out because it's critical. This is just a chemical lab in here. This is chemistry has to happen, so it has to be protected. If you have stray elements coming in here, it can interfere with the chemistry that's happening in here. This chemistry continues for the lifetime of the cell, and in some cells that's your entire lifetime. So it has to be controlled very, 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 very carefully. So I hope this helps.